composition of the enzymes that are present in pancreatic juice, their actions. The bicarbonate ions that are released, its actions. Mechanism of synthesis of pancreatic juice and regulation of pancreatic secretion. Okay? So, if we get a section of, histological section of pancreas, it will look like this. So, at the center, we know pancreas is a mixed organ. It is endocrine as well as exocrine. So at the center, this part is the endocrine part. This part is called islets of Langerhans. Islets of Langerhans. Thank you. You will get different types of cells here. Alpha cells, gamma cells, delta cells and other cells. And since it is endocrine, islets of Langerhans are endocrine in nature, so definitely they will be secreting hormones. They will be secreting hormones. And these hormones are insulin, glucagon, somatostatin. Okay? So these are the hormones secreted from the islets of Langerhans. That is the endocrine part of pancreas. But this is not the point of our discussion. Our point of discussion is this part. These are the exocrine part. If you look at this part, you will find there are some acinas. Lot of acini. Acinas is singular and in plural it is acini or acini. Okay. So within this there are tubular structures. The secretions of these acinas, the acinar cells are lined. So their apices, means apical part, they will be secreting enzymes. These cells, this apical side of the acinar cells will secrete enzymes into the lumen. These enzymes will be carried through the ducts. Initially by intercalated ducts, we'll see this. So this is one of the acinas. So acini, these are acinis. So these are acinar cells. And within these cells, you will find these granules, Zymogen granules. These Zymogen granules are actually the enzymes, proenzymes, precursor of enzymes. These are precursor of enzymes. We'll come into this. What are the different precursor of enzymes? What are these? So they are actually responsible for the synthesis of different pancreatic enzymes. Which cells? Acinar cells. So their secretion, so they will secrete these contents through exocytosis into this lumen. You know exocytosis? This is the basal part, the base of the cells. This is the basolateral aspect, and this is the apical side. So these granules are actually synthesized here. Cells, there will be nucleus. Within the nucleus, there are DNAs, means genes. They will be producing mRNAs for different proteins. Here protein means enzymes. All enzymes are really, almost all the enzymes are protein in nature. So they will be synthesized initially in the endoplasmic reticulum. So these cells will be having endoplasmic reticulum. After their synthesis, they will be going to their Golgi bodies. There it will be packaged, and they will pour these Golgi bodies will produce granules, vesicles containing these enzymes. And when the cell is stimulated, when these are ready with secretions, when these cells are stimulated, these granules migrate to the apical aspect. To the apical aspect, see, that's why all these granules are gathered here. They are not gathered here. They are ready to secrete into the duct. So when they will be receiving any stimulus, immediately these granules will be exocytosed into the lumen. Then they will be carried through this. While passing through this, they will be facing these ducts, intercalated ducts, these ducts, interlobular ducts. So they are lined by some cells. These are ductal cells, duct cells. These are also very important. These duct cells are also very important. They are also innervated by nerves. They are having also receptors for different ligands. What are ligands? Ligands are substances that act on a cell, but they cannot enter within the cell to show their activity. So what they will do? These ligands will be having receptors on the surface of the cells. They have got their specific receptors. So when these ligands bind with these receptors are present at the surface of the cells, basolateral surface of the cells, not in the apical region. So these receptors are present. So when these ligands bind, these ligands are nothing but hormone. It may be hormones secreted by some other organ, or it may be the local hormones. So they will bind with this receptor. These ligands, gastrin, ligand, secretin, ligand, cholecystokinin, pancreazymin, these are ligands, serotonin, histamine, these are ligands. So they will bind to their respective binding sites, receptors. When they bind, immediately they produce some second messenger within the cell, because the other side of the receptors are having some enzymes connected to these receptors. Their activity is increased with the binding of the ligands. So those the enzymes inside the receptor, they will be activated and they will produce second messenger like cyclic AMP, calcium ion. Okay, this sort of IP3, though this sort of second messengers will be produced, and these second messengers will make necessary change within the cells. Okay, so change means it will initiate production of enzymes. It will initiate within the cell, it will stimulate production of the enzyme, means mRNA, means activity of the endoplasmic reticulum will be reduced, so they will be producing proteins, and these proteins will be processed in the Golgi apparatus. Then they will be membrane bound vesicles, and when the cells are stimulated, immediately they will be exocytosed into the lumen. Okay, so ligands can cause this, or the nerve, vagus nerve. From the nerve ending of vagus nerve, there is secretion of acetylcholine. Now this acetylcholine will bind with their corresponding receptors present on the basolateral surface. When they bind, immediately calcium ion is released. So this calcium ion will cause initiate exocytosis of these granules. So there will be secretion of enzymes. So if you stimulate vagus, what will be there? Secretion of enzymes. These secretions, so these secretions are enzyme rich. But when they will be passing through these basal cells, these duct cells will secrete bicarbonates, will secrete a fluid containing rich in bicarbonate ion, means this fluid will be alkaline. See the purpose of pancreatic juice. The food bolus has to enter from here to here, but this food bolus is having lot of hydrochloric acid. So this food bolus is actually acidic. These are actually acidic. But if these bolus come here regularly, with the contact of this acid, the duodenum will face ulceration. There will be duodenal ulcer. There will be inflammation of the duodenum. To prevent that, the bicarbonate ion should be alkaline, fluid should be there. So they will neutralize this excess hydrochloric acid. So in presence of acidic food bolus in the duodenum, just they are going to enter. Immediately the wall of the intestine will sense it. The cells present in the wall of the intestinal wall, there are some cells, they sense this is acidic. So this reflexly will secrete bicarbonate rich fluid from pancreas. And this is done through secretin. Reflexly through a local hormone, secretin. These secretin are secreted by argentafin cells or S cells of the intestinal mucosa. Tomorrow we will study this. Okay, so there are some cells, S cells or argentafin cells, they will be secreting in presence of hydrochloric acid, food rich in acid, they will be stimulated and they will produce this hormone. What is this hormone? Secretin. Now this secretin will be circulated through blood. It will enter into the blood, and this blood through circulation will be coming to pancreas, will be coming to the duct cells, and here they will cause stimulation of the duct cells to produce more bicarbonate, more acidity, more bicarbonate. So purpose is to neutralize the excess acid and to make the medium alkaline, because all these enzymes will be acting in alkaline medium. So medium is ready and hydrochloric acid is neutralized. Okay? These cells, as I said, the basal cells, the duct cells, as well as these cells, acinar cells, they are innervated by sympathetic as well as parasympathetic nerves. So their nerve endings will secrete, parasympathetic nerve endings will produce acetylcholine. So they will initiate secretion, and this secretion will be enzyme rich. Whereas the sympathetic stimulation will inhibit the secretion of pancreatic juice. So sympathetic stimulation will cause reduction in secretion of pancreatic juice, and parasympathetic stimulation is always stimulatory. In heart it is inhibitory. Parasympathetic stimulation, vagus nerve stimulation will reduce cardiac activity. But we can see in digestive glands, they have got opposite. They are stimulating in nature. Clear? So these are the acinar cells. They will be secreting enzymes, different types of enzymes. These are the Zymogen granules, and this is the nucleus of the cells, highly active. And these are the duct cells. And some duct cells, these are duct cells. Some ducted cells will be present within the acinars. They are called centroacinar cells. These are called centroacinar cells. They will also, these though they are present within the acinars, they will be producing bicarbonate ions only. They will be producing bicarbonate. When they will be stimulated, they will be producing bicarbonate. When there is acidic food within the duodenum, the wall, intestinal wall is having I cells. There are some cells. They will be stimulated by this acid and will cause secretion of secretin. This secretin will be circulated into the blood and ultimately will return to the pancreas, and here it will stimulate the centroacinar cells or the duct cells, and here they will produce bicarbonate ions. And with this bicarbonate ion, they will be neutralizing the excess acid. So these are intercalated duct. Now these intercalated ducts will join this duct. It's an intralobular duct. So there are different lobules. All these lobules will be joining together and will form a duct. A common duct. Duct of, what is this duct? Duct of Wilson. This duct. This is the pancreatic duct. Here it will be divided. One part will go there. This is the main pancreatic duct. Through duct of Wilson, it is entering within the duodenum. Through a sphincter, there will be a sphincter of OD, and this inflated part is called the beta, ampulla of beta. So through this ampulla of beta, the pancreatic juice will be entering within the duodenum. Okay, and you can see this duct is divided, and it has formed another duct. This duct is called duct of Santorini. This Santorini opens into duodenum, but about 2 centimeters above. But its role, secretion of this part is very less. The main part is through ampulla of beta. Now you just see what this is. What? Gallbladder. There is, by some means, because of inflammation or some other reason, some stones are formed there. Now these stones, from this through cystic duct will enter into the common bile duct, and by the pressure of the bile it will proceed toward this and proceed. If the stone is a little larger, then they will be stored here, causing obstruction of bile flow. From here what is coming? Used to come bile, and from this pancreatic juice. So now because of this obstruction here by the gallstone, the pancreatic juice will be unable to release here, bile will also be unable to release here. There will be tremendous pain. And if it continues, there will be jaundice. Because bile will be released into the blood. And bile contains pigments, bilirubin. So because of presence of bilirubin, the mucosa will be yellowish. skin will be yellowish, icterus will be there. Okay? Because of obstruction. This is called obstructive jaundice. Okay? Not only this, the patient will suffer a severe condition, may die also. What is this? Pancreatitis. Because the juice will be unable to, pancreatic juice will also be unable to pass through. So there will be the pancreatic juice will be unable to come. So there will be some changes. The pancreas will become inflamed. The cells will be destroyed. Because during this time, two components of gastric juice becomes activated. One component is phospholipase. It was inactive, phospholipase A2. But in such condition, this phospholipase A2 becomes activated. As a result, these activated enzymes locally here will be activated here. Activation, because of activation, it will produce lysolecithin. Lecithin is a phospholipid. So it will produce lysolecithin. The main function of the A2 was digestion of phospholipid within the intestinal canal. But here, because of inflammation or some other reason, this phospholipase A2 become activated. As a result, it will act on, it will convert phospholipase, prophospholipase, inactive phospholipase A2 into active. Soon it will become, as soon as it become, it will be digesting, it will be, since it was activated within the pancreas, it will be digesting the fats present in the pancreas. The fats there are structural fats there, and these are made up of lecithin. So immediately it will convert this lecithin into lysolecithin. Lyso means breakdown. Lyse. So this lysolecithin will break down the cells, the fatty components. Thus, there will be necrosis of the pancreatic cell. And moreover, within the pancreatic juice, within the pancreatic, there is another component called antitrypsin. See, trypsin is very important. The pancreatic juice contains several proteolytic enzymes, and all these proteolytic enzymes are inactive. Since they are inactive, they are unable to digest any protein in the pancreas. So, for this reason, they are unable to destroy itself, since they are inactive. But if by some any means, trypsinogen, inactive form, becomes trypsin, then it will be starting digesting its own cells. So it will be then self-destructive. It will destruct the pancreas itself, and it is a life-threatening condition. The patient may die because of hemorrhage, because it will also destroy the blood vessels. So two components. One is inactivation of antitrypsin, activation of trypsin, and activation of phospholipase A2. It happens during pancreatitis. When somebody is suffering from pancreatitis, immediately we think his pancreatic cells are dying. So if his condition is severe, he may die. It may happen those who frequently take alcohols. Because of alcohol abuse, pancreatitis may happen. Somebody punched here. So here your pancreas may be damaged, injury. So that will also cause activation of those enzymes and the self-destructive process will begin. So one cause of pancreatitis is obstruction created by gallstone. So there are some reasons of pancreatitis. So this is important for clinical questions. Okay. One thing, remember, pancreatic juice composition of this is wrong. Why wrong? Trypsin, you won't find trypsin in pancreatic juice. It will be pepsinogen. So while you will be writing, all these are inactive. Is that clear to you? So there may be short question. Among the following, which one of these is not present in, you will find trypsin, the name trypsin is there. And others are pro chymotrypsin, but in case of trypsin, it is simply written as trypsin. But this is wrong. It will be trypsinogen. Okay, so that's why I have written there. Most of these enzymes are inactive proenzymes. When they will be entering within the gastric, within the pancreas, within the intestinal lumen, first one thing will be activated. Out of all these enzymes, when this secretion, there will be secretion through the ampullar bladder into that intestinal lumen, into the duodenum. First, one of the enzyme will be, proenzyme will be activated, and it is who? Trypsinogen. And this trypsinogen will be converted into trypsin, then this trypsin will make all other proenzymes, inactive enzymes, into their active form. So this will be happening within the lumen of intestine after their secretion, not before their secretion. Who is doing this activation? Trypsin. Now how the trypsin is activated first? The enzymes, these are all proteolytic enzymes. Elastase, proelastase, digest what? Elastin. Collagenase, collagen. What are these? Elastin, collagen. Whose skin is tight? Mine or yours? Yours. Why? Under the skin, you are having lot of collagen, lot of elastin. I am losing. That's why my skin is loose. Same thing will happen to you. Okay. See, there is another type, nuclease, ribonuclease. You are eating flesh. Okay? This flesh means cells. Cells means in each cell there will be DNA, RNA. So now DNA, RNA, this compound needs to be digested. These are digested by DNAs, RNAs, ribonuclease, deoxyribonuclease into nucleotides. Okay? And carboxypeptidase. Now you see carboxypeptidase, these are amino acids linked together by peptide bonds. All the amino acids are having at least one carboxylic group and one amino group. At least one carboxylic group. Well, she is the one of the amino acids. She is one of the amino acids. So this is her carboxylic group and this is her amino group. Okay? Everybody's right hand is carboxylic group, left hand is amino group. So we are going to form a peptide linkage, peptide bond or peptides. Numbers of amino acid will be linked together by peptide bonds. So what will be there? Her free, no, nobody is there, and she will bind with this through a peptide bond, amino group, again her carboxylic. Amino carboxylic, and now there is a peptide bond. So now this carboxypeptidase, where it will act? It will look, where is the carboxylic group is free? Oh, it is here. So I will break this. You are having one peptide bond? I will break this, you will be free. I can break this also. I will start breaking from this end, not from that is amino terminal. That is amino terminal. So this enzyme will be starting the breakdown of, if it breaks this, so there will be one dipeptide. If it breaks this one, there will be one tripeptide. And if you break this is short peptides. So if you suppose there are 5, so I have broken this, 2 dipeptide and tripeptide. Then again if I break this, amino acids and dipeptide. Then again I break this, 2 amino acids. So this way, this carboxypeptidase, particularly the R group, this may be aliphatic group of the amino acids, this may be aromatic group of the amino acids. So the aliphatic aromatic group, the amino acids who are having from this end, who will be having aromatic or aliphatic group, that peptide bonds will be broken. Is that clear to you now? Sure? Okay, that's good. So what are there? Next, chymotrypsin, trypsin. So these are the proteins we are coming to, and there are amylolytic enzyme. Amylolytic enzyme means carbohydrate splitting enzymes. It is very similar to this. This means salivary amylase. It will break only alpha 1,4 glycosidic linkages. Not the branching enzyme, branching bond, which is 1,6. There may be 1,2 glycosidic bond. So it will not act on that. For that, different enzymes are there. Okay, they will break. Ultimately, everything will be broken. Ultimately, you will get glucose, lot of glucose. How does it differ? Question comes. How does it differ from salivary amylase? Both are acting by breakdown of hydrolyzing the alpha 1,4 glycosidic. Now how they differ? How they differ? Short questions or MCQs. See the pH here and the pH there. Where it is more alkaline. So this amylase can act at more alkaline range, but here it cannot. 7 to 11, but it can act in it acts in alkaline medium, 6.6 to 7 this pH, or 4, starting from 4, it can act, no, not 4, 4 is acidic. So when this amylase enters into the stomach, it will face acidity. So as acidity increases, the salivary amylase loses its activity. The pH range through which its act is very narrow, but it is very wider. Okay, and another difference. Salivary amylase cannot digest unboiled rice, cannot digest unboiled starch. But if you take unboiled rice, no, ultimately this will be, because this will involve, intact rice will not be coming out, because these are digested. So, digested by pancreatic amylase. They are able to digest starch which are not boiled. These two are the difference between the two. You will face the question. Pancreatic lipase, action of lipase, digestion of fat, triglyceride. Sorry. This is glycerol. This is what? All. This is one alcoholic group, this is another alcoholic group, this is another alcoholic group. Together with this alcoholic group, together with this alcoholic group. When an alcohol reacts with an acid, what is formed? Ester. Ester. Acid and alcohol, when binds together, forms an ester. So, fatty acids, different types. Number of carbon acids may vary. It may be long, it may be short, depending on the number of carbon atoms. It may vary in whether these carbons are saturated or unsaturated. If there is unsaturated double bond in between the two carbons, this is unsaturated. These are valuable for us, because this type of unsaturated fatty acids are not synthesized in our body because of lack of these enzymes. Who will be converting, you will be doing this. So that's why they are, but they are very important. So we have to procure them. We have to take those fatty acids because these are structural component. As we have seen in the cell membrane, there is phospholipids, and you see there are two legs, no? In phospholipid molecule, these legs are of fatty acids, which are like this. These are long chain fatty acids, which are straight. These are short chain fatty acids. Okay, among these fatty acids, some are unsaturated. So if there is, we know, depending on the saturation and unsaturation, the flexibility of the membrane will depend. Presence of cholesterol will determine the fluidity of the membrane. So these are important. So this important has to be procured from outside through other oils, outside oils. That's why these fatty acids are called essential fatty acid. Linoleic acid, linolenic acid, alpha-ketoglutarate acid. These are essential fatty acids. Okay. All these fatty acids are having carboxylic group. R means rest of the carbons of fatty acid. So they will form a ester bond after liberating water. So they will be linked by, so one fatty acid molecule join glycerol. So what will be the product? Monoglyceride. Monoglyceride. So in this way, another fatty acid can also join. Now it will be diglyceride. When another molecule joins this, it will be triglyceride. These are fat. So this pancreatic lipase, with the help of co-lipase, with the help of bile salts as their cofactor, they will hydrolyze these bonds. One fatty acid will be separated. And what I will get? Diglyceride. If another fatty acid, another bond is hydrolyzed, now how I will get? Monoglyceride and one molecule of fatty acid. If I break this, I will get glycerol and another molecule of fatty acids. Understood? Clear? But when diglyceride is formed, fatty acid, one diglyceride, one fatty acid will be formed by the lipase. Then one monoglyceride and another fatty acid will be formed. Lipase will be okay, done. Enough. Why enough? Monoglycerides and the fatty acids, these monoglyceride and fatty acids now can be absorbed. No need of further. If you can, okay, no problem. But if you cannot digest the monosaccharide further, no issue. This will be absorbed as such. Okay? Very good. Everybody is attentive, I think. Cholesterol ester. Now cholesterol, all means there is a alcoholic group. Alcoholic group. So it can also bind with fatty acid through ester bond. So to break this bond, cholesterol ester bond, there is one enzyme. Cholesterol esterase. We are eating lot of cholesterol through milk, through egg, through fat. So that has to be digested, and that will, this cholesterol, some cholesterol, these are always in conjugated form as ester form. So that should be, as a result, free cholesterol will be there. Now cholesterol will be absorbed, and the fatty acid liberated, this will be absorbed separately. Done? Okay. Then we got phospholipase A. So phospholipase A, it hydrolyzes phospholipids, lecithin, lysolecithin, kefalin, sphingosine. So these are the phospholipids. So they will take away one fatty acid from them, and it will be lysophospholipid. And one lysophospholipid is dangerous. I already told you. What is that? Lecithin. So this will destroy, this is destructive in nature. It will cause necrosis of the cell. And next to this, phospholipase B. This also hydrolyzes phospholipids, same manner. And co-lipase, these are actually coenzyme supporting the lipase. Without the support of co-lipase, it cannot act. Not writing anything? Right. Bile salt activated lipase. Some lipases they are there, they will be activated by the bile salts only. Where from this bile salt will come? Through bile. This bile is stored in gallbladder. When you will be taking food, gallbladder will be constricted. So this bile will be coming through into the duodenum along with pancreatic juice. So in the pancreatic juice there is lipase, and in the bile there is bile salts. So they will join together and this lipase will be activated and it will start its action. Action is same. Another important constituent is trypsin inhibitor. It is not written there. So this trypsin inhibitor prevents conversion of trypsinogen into trypsin when it is within the pancreas. In absence of trypsin inhibitor, there will not be any inhibition. So now anybody can convert trypsinogen into trypsin within the pancreas, like inflammation. So as a result, will be the trypsin once activated within the pancreas, it will start digesting, thinking, this cells is nothing but it's like meat, flesh, so digest it. You have seen spider. What happens to the mother spider? When they will be having eggs, no? When from these eggs within this, these are generated, they will start eating their mother. So here also when trypsin, if by any means becomes activated, they will be eating their mother cells. Very sad. So you should be very careful. Don't eat, don't drink. If I don't drink, how can you be modern? No? What are you talking, sir? This albumin, very less amount of albumin you will find, and these are the bicarbonates. Mostly bicarbonate. So this part is mostly these cations are very less, but these anions, among these anions, this concentration is very high, and it varies from 110 to 150. When under resting condition, I am not eating anything, this content of bicarbonate is less in near about 110. When you are eating something, you are eating food, then its secretion of bicarbonate is increased and the level may rise to 150 milli equivalent per liter. So all the function I have mentioned. Yes, left, I'll tell. Skin. See. Epidermis. And this is the dermis. What is there? And that's why skin is, you can move it here and there, so you can slide it. Because, so since there is less collagen, less elastic, I can move more, but yours one is tight. See how these are arranged? See, these are collagen, and this green, these are elastin. They are tightly holding like a knot, so that, don't sleep, I am there, you cannot go apart, like net. So these are the functions of elastin. So we are eating flesh, no? So we have to digest this elastin. We have to digest this collagen. From this collagen digestion, we actually get gelatin. In cooking, we need gelatin to make it. Very. So for digestion of this collagen, there is collagenase enzyme. For digestion of gelatin, there is gelatinase enzyme. For digestion of elastin, elastase enzyme is there. Learn this, learn this. What is gelatin? Learn. Vertebral column. There are segments, no? In between the segments, what is there? What is there? And disc. Disc is there. And if this is slipped to a particular side, we call. So next part in the classroom before practical. Okay.